Okay, now we go on to question number 15, which is talking about the checkpoints in regulating the cell cycle. So, figure 11 on page 231. So, here it is going, like, there are checkpoints as the cell cycle continues, and these checkpoints ensure that the cell cycle is going on correctly, and they monitor the cell cycle. So if the cell cycle, it has built-in checkpoints, if and that is monitoring the cycle, and it can stop if anything is going wrong. For example, a checkpoint is there near G1, and this is monitoring that is the DNA damaged. If the DNA is damaged, the cell cycle will not continue. Not damaged, it will continue. It will go on to the next. So there are other quality checkpoints at stage S where DNA replication is taking place. Is it going on well? And at G2, that is it ready for mitosis? So all these checkpoints, they are monitoring the progress of the cell cycle. So here it says cells use, and how do they check? What are the checkpoints? Like, what do they have? So they use cyclins and cyclin dependent kinases. This is what is used. These are proteins, cyclins, and cyclin dependent kinases are the enzymes. So they are using a protein, cyclin, and an enzyme, cyclin dependent kinases, which are also called CDKs. They are used to control the cell cycle. So they combine. So the combination of cyclin and CDK, they start the cell cycle at different checkpoints and monitor the cycle for quality control in the G1 stage. Here, then the cell is growing and preparing to replicate DNA, everything good. Then DNA replication occurs in the S stage. Then in the G2 stage, the cell takes again an inventory if everything has is ready. Is it ready to go for mitosis? If the spindle apparatus malfunction is there or no, then it will. If there is a malfunction not working properly, then the cycle will not proceed to cytokinesis. So there is a checkpoint at mitosis. So the cell cycle in eukaryotic cells is driven by a combination of two substances. This is important. One is cyclin and the other is cyclin dependent kinases, CDK. And this combination, cyclin side CDK, they control the activities. So this is it. These are the questions based on it. Where does, according to the overview of checkpoints, during which phase does the cell cycle evaluate if the DNA is damaged? So that is the G1 because it's still growing. What is the primary purpose of quality control checkpoints? Why? So that they monitor the cell cycle. And if it is going wrong, then it stops. Cell cycle, you get, is driven by a combination of what two substances? Cyclin, which is a protein, and cyclin dependent kinases, enzyme. So these are the questions. These are some more questions. Now we come to question number 60. Here you have to talk about cancerous and non-cancerous cells. What do you know about cancerous cells and non-cancerous cells? So this is a figure. This is a normal cell, and this is cancer cells. They're dividing, so much. Cancer cells often have an abnormal, irregular shape compared to normal cells. See the shape here. In this stage, in this image, some cancer cells are entering vessels which might carry them to another part of the. See, they are entering here. They can take it to other part of the body. This is one way cancer can spread from one body part to another. So, what happens in cancer? When cells do not respond to the normal cell cycle control, that is the checkpoints, they are not responding to the checkpoints, then a condition appears which is cancer. Cancer is uncontrolled growth and division of cells. A failure in the regulation of because cell checkpoints are not working. So growth is going on, cell division is going on. When unchecked, cancer cells can kill an organism by crowding out normal cells, resulting in the loss of tissue function. So they crowd out the normal cells and they can be a loss of function. Cancer cells spend less time in interphase than do normal. In normal cells the interphase is the longest phase, but in cancer cells they spend less time here, which means cancer cells grow and divide unrestrained as long as they are supplied with nutrients. Now, this is how they can intrude the normal cells. Cancer is uncontrolled growth and division of cells. It is the result of failure of regulation of cell cycle. They lose control when genes that regulate the cell cycle are damaged. So, when does it happen? When the genes are damaged. Okay, and that is uncontrolled growth of the cell. The same thing. So non-cancerous divide at control rate, follow such checkpoints, stop dividing when cells are present or when they are not in contact with the neighboring cells. When they come in contact with they help in normal growth and repair of tissues. Cancerous, fast, uncontrolled, ignore signals, continue dividing even when new cells are not needed, and they form a mass of cells which is called a tumor. A tumor forms and then it becomes cancerous. What is the inference about it? The cancer cells have higher rate of cell division than normal non-cancerous. Normal cells divide only when it is required. Cancer cells divide continuously and there is abnormal growth. Now here are the questions. Pause the video and answer the question. Characteristic of cancer cells, they contain multiple genetic changes. Then the ultraviolet radiation is an example of a carcinogen. Carcinogen are those that cause cancer. A failure is the regulation of the cell cycle will result in cancer. When carcinogens cause a failure in the regulation of cell growth and development, what condition happens? Cancer is formed. This is substances known to cause cancer are carcinogens. We call them carcinogens. They cause cancer. So UV is one. You know, UV can cause skin cancer. So it is one carcinogen. More questions? Question number 17. Recognize that each zygote receives half the number of chromosomes from the maternal gamete and the other from paternal. So here, you know that there are two types of cells. We can say haploid and diploid. Haploid have half the number of chromosomes than a normal cell, and diploid has the normal cell. So we say that haploid has n number of chromosomes and diploid has 2n number of chromosomes. So they maintain the same chromosome number. They produce, organisms produce gametes. Gametes are sex cells. They have half the number of chromosomes. Humans, each gamete contains 23 chromosomes, and N is used to represent the number of chromosomes in a gamete. So whatever is the number of chromosomes in a gamete, we call it N. So which has N chromosomes, it is a haploid cell. It means it's haploid means actually single. The process by which one haploid gamete combines with another is called fertilization. As a result, the two ends are going to combine and it will have two N chromosomes. So the zygote that is formed after fertilization will have two N chromosomes, and this is called a diploid cell. So you should know what is haploid and diploid. Here you can see the individual on the left has an X chromosome and a Y chromosome. This individual is male if the individual has both X chromosomes, then it will be a female. So females have both X chromosomes and males have one X chromosome and one in the 23rd pair of chromosome, that is the sex chromosome. So we have 23 pairs. Out of these 23 pairs, 22 pairs are the same in both males and females. The last pair, 23rd pair, which is called the sex chromosomes, they can be different. There are two types. They could be X and Y. So individuals, two X chromosomes, they are genetically classified as females. Those with one X and one Y are males, and both, and that's like, this is the sex chromosome. So the pair of sex chromosomes which you have. One is coming from the female, one is coming from the male. Okay. So here there are questions based on this. An individual has two X chromosomes. What is their gender? Female. Which chromosome is present only in the male? That is Y. How many pairs all 23 pairs of chromosomes are autosomes. So 22 are autosomes, and the third pair, the last 23rd, it is the sex chromosome. So we have sex chromosomes and the autosomes. Okay, so autosomes are the normal, like, which are the same in both males and females, and the 23rd pair is sex chromosomes. So this XX will be female, XY will be male, YY is not possible. So this is it. Now question number 18 talks about meiosis. How does genetic variation takes place? Figure 17. This is figure 17. So here we'll talk about homologous chromosome. Now what are homologous chromosomes? Each parent contributes 23 chromosomes. We studied the chromosomes that make up a pair. One chromosome is from each parent. So when we are saying pairs, 23 pairs, it means one is from one parent and one is from the other parent. So this is a pair of chromosomes. So they are called homologous chromosomes. Homologous chromosomes in body cells have the same length and the same centromere position. They have the same position, centromere, and the same length, and they carry genes that control the same traits. So these are homologous chromosomes. Centromere the same position, and these are and they can be divided into sister chromatids. So here you can see these are the homologous chromosomes. Now what's happening here? Crossing over is taking place in the sister chromatids, and they exchange parts. Now they have exchanged the parts, so the traits have changed now. So they are no longer like this, they have changed their parts. So the red and green chromosomes have exchanged segments. This exchange occurs during synapses, that is crossing over. It's the process during which the chromosome segments are exchanged between pair of homologous chromosomes. And because of this, there can be genetic variation. That's why there is variation. They are not identical. Now, this is the meiosis, the whole meiosis. You have to remember meiosis. Go through this chart carefully. Meiosis can be divided into two main phases. One is meiosis 1, this is meiosis 1, and then meiosis 2. After meiosis completes, there are four cells that are produced. So gametes are formed during the process called meiosis. Here, this happens in the sex reproductive organs, and whether the different cells are being formed, reproductive cells are being formed. So in this type, reduction is taking place. It is reducing the number of chromosomes. In mitosis, the number was not reducing. Here, the number is reducing. It is becoming haploid. It is referred to as reduction division. So, meiosis is also called reduction division. Supposing they give you a question and they don't mention meiosis, they say reduction division. You should understand that it is meiosis. Meiosis occurs in the reproductive structure of organisms, and they form haploid gametes or spores. While mitosis maintains chromosome number, cellular reproduction, and in organisms that reproduce asexually, meiosis reduces the chromosome number. So mitosis is maintaining it, meiosis is reducing it to half. A cell with 2n number of chromosomes will have gametes with n number of chromosomes after meiosis. This is it has two cell divisions. One is meiosis 1 and meiosis 2. Meiosis only halfway completed at the end of meiosis 1. Now this is similar to mitosis, just like mitosis we've already studied. The prophase 1 is similar to the prophase of mitosis, then metaphase 1, anaphase, and here telo. But only what's happened, crossing over has taken place. You see, there is variation. So crossing over is taking place here. So in prophase 1, the pairing of the homologous chromosomes take place, and chromosomes consist of two chromatids, sister chromatids. Then crossing over starts here. So that happens in meiosis 1. During prophase 2, a second set of phase begins. The spindle apparatus forms and the chromosome condenses. So second phase will start here in prophase 2. Now there are two cells. In meiosis there was one cell, here there are two cells. Now after telophase, during metaphase 2 the chromosomes are at the middle in the equator. During meiosis metaphase 2, a haploid number of chromosomes line up at the equator. They separate and they line up here at the equator. During anaphase 2, a sister chromatids are pulled apart. Now here the sister chromatids are pulled apart. At the spindle fibers, the sister chromatid move towards the opposite poles, and then the telophase, the same thing, and then the cytokinesis, and four cells are formed. So let's go through it. Okay. So this is meiosis 1. Interphase, chromosomes replicate, chromatin condenses. In prophase 1, they pair together homologous chromosomes and crossing over starts taking place. Then here the nuclear envelope will break down and spindle will form. Metaphors, they line up and they attach the same like mitosis, attached to the centromeres. Anaphase 1, the homologous chromosomes separate and move to the opposite poles. Then meiosis 2, the prophase 2, this is telophase 1, the nucleus again forms, spindle breaks down, chromosomes uncoil, two nuclei are formed and the cell divides. Then prophase 2, chromosome condense again, spindles form in each new cell, spindle fibers attached to the chromosomes, they line up near the equator, and then anaphase 2 they separate, centromere split, sister chromatids, and then here you can see they are having telophase, four nuclei form around chromosomes, and the spindle breaks down, and the products are four cells, and they each have half the number of chromosomes, which is haploid number of chromosomes. This is meiosis. Here they have given you the description of all the phases, which number represented prophase 2. So you can see in meiosis 2, always you are seeing like, two cells, two cells. Here you have single cell. So phase 6 is prophase 2. These are the questions based on that. All questions are based on that. Meiosis. Many questions here. Too many, you can just like pause and have a look. Then we come to question number 20. Karyotype. What is karyotype and how is it used to study genetic disorders? So this is the male karyotype, because you can see here you are getting the XY chromosome. So this is male and here this is female. All same except the last one, which is XX. So karyotype is the study of genetic material. It does not involve study of genes alone. They study whole chromosomes by using images of chromosomes. They stain the chromosome and they see the whole images. That is called a karyotype. And when do they do it? During metaphase. The staining bands identify or mark identical places on homologous chromosomes. During metaphase of mitosis, each chromosome has condensed greatly and consists of two sister chromatids. So you can see every chromosome has two sister chromatids. The pairs of homologous chromosomes are arranged by decreasing size. When we are arranging them in decreasing size. You can see the big ones here and the small. This micrograph is called a karyotype. Karyotypes of a human, female and male, are shown here. So if they give you this figure and say, what is it? It's a karyotype. They might say, which one is a male? So you will see the last one. If you have XY chromosomes, you will say male. XX, you will say female. Then what is non-disjunction? During cell division, the chromosomes is separate with one another of the sister chromatids going to the opposite poles. So new cells are formed. Cell division sometimes during which sister chromatids fail to separate. They're not separating properly. This happens sometimes. This is called non-disjunction. Separation is not taking place properly. If non-disjunction occurs during meiosis 1 or meiosis 2, the gametes will not have the correct number of chromosomes. When one of these gametes fertilizes another gamete, the resulting offspring will not have the correct number of chromosomes. So, non-disjunction occurs. If there is a set having a set of three chromosomes of one kind is called trisomy. So if there are three chromosomes, it's trisomy. If there is one chromosome, then it's monosomy. So if you see this figure here, it will be better. This is non-disjunction in meiosis 1 and meiosis 2. So here you see when the division is taking place, somehow there is this happens. The chromosome do not come here. And this is now both are here. So the division which will take place here does not have any chromosome. And then when it is fertilized, it will have only one which is coming from the sperm. So it will be monosomy. Now here both were there. Then when after fertilization it becomes three. So it is trisomy. So this happened because of non. In meiosis 2, what happens? They should divide like this. They divide, then they separate. But when they are separating, instead of going to the other one in the eggs, it goes here. Both are here. Now when fertilization takes place, this one has only one, monosomy and trisomy. So this is non-disjunction. Autosomes are chromosomes that are not sex chromosomes. Humans have 22 pairs of chromosomes. Down syndrome, Patau syndrome, and Edwards syndrome are all examples of non-disjunction in autosomes. When it is taking place in autosomes, Down syndrome happens, Patau and Edwards. One of the earliest known human chromosomal disorder is Down syndrome. You all have heard about it. It is when there is an extra chromosome 21. So when there is an extra chromosome 21, there is Down syndrome. So often it is called trisomy 21 also. We also call it trisomy 21 because there is one extra chromosome in this. So here this table is important. If you have like XX, then it will be genetically classified as female. XO, that is only one X, it will be a female with Turner syndrome. If there are three, no phenotypic effect. Like this, they are males because there is one Y. Male with Klinefelter syndrome, there are two X's, one Y. One X, two Ys, no phenotypic, results in death. So, non disjunction occurs in both autosomes and sex chromosomes. Some of the non disjunction in human sex chromosomes are shown here. This go through this table properly. Klinefelters and Turner and that. And Down syndrome is actually in autosomes. Now here they've given you a question. Mysis one. These are some questions. Okay, so we will do till here. Next we'll continue in the next video. Okay.